Okay, I'd like to welcome everybody to the June 9th meeting of the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics. As you know, our remit is actually much broader than the name of the working group might suggest. And so today we're going to be venturing off into some very interesting areas of modeling. I have to remind you, as always, that this meeting is live streamed and recorded, so please remember that as part of your discussion. As always, Tom and I are available for suggestions, comments, criticisms. Jim Sluka and Lorenzo are also usually here to help out. They're always here online if you need them. Jim in particular for anything related to the IMAG wiki. We have our usual communication channels. They work better if you use them. We have MSM Viral as a Twitter feed. We have the IMAG MSM wiki pages. Please use those to advertise your jobs, to publicize your papers and talks. Anything that you think is relevant to the working group that you would like to see there, please put up. We have a LinkedIn page for the IMAG MSM as well. And we have our YouTube channel with our growing archive of great talks. So please do help us distribute that information and make sure that people take advantage of it. Again, we do have this Twitter hashtag / hash MSM Viral. If you tag things related to the working group with that, it helps get the word out. You could retweet the announcements if you like. That would also be very helpful. Are there any announcements before we start the seminar today? We have a steering committee meeting tomorrow morning, Friday, 11 a.m. EDT. If you have an invitation already, we hope to see you tomorrow. If you think you should have had the invitation and you didn't receive one, please do let us know and we'll try to make sure that you are on the list for that invite. Next week, we have a slightly different program from usual. People will remember Claire Bryant from the great talk she gave about a year ago for the working group. She's going to be visiting in the United States and giving a virtual seminar as part of that. And she's agreed to let us use that virtual seminar a week from today as a special seminar for the working group. Note that the Zoom information for that seminar is slightly different from our regular one because it's a shared seminar. The Zoom information will be sent out again by Lorenzo, and I'll also put it in the chat before we're done today. I realize for our European friends that a 6:30 p.m. US time is not very convenient. 11:30 in the evening in England, 12:30 in the morning in the mainland. I apologize for that, but that was a limitation. As usual, the meeting will be broadcast and recorded though, so if people want to hear Claire's talk, follow up for her earlier talk, you'd be welcome to listen to it after the fact if the time is bad. I guess at least for people on the West Coast and maybe in Asia, it's not so bad for time. After that, we will have Fiona McFarland from St. Andrews University, whom we've wanted to have speak for some time. She'll return us to our regular slot in the afternoon. And we do now have this very convenient Google form that allows you to suggest speakers. Please do fill that out. It puts your suggestions into a table. Lorenzo and I and Tom go through that table every week and send out invites. So we've tried to make it as easy as possible for you to suggest the people you would like to hear speak. So please help us out with that. Okay, well, I don't want to take any more time from our speaker today. We're going to be hearing about modeling of cancer evolution in the context of Boolean network models. And I am very excited that Laurence has agreed to speak to us today. So I will turn over the screen share at the meeting to you.
Thank you very much. Let me share my screen, make sure everything is okay before I thank you for inviting me. Is it okay? Okay. So again, yes, thank you for the invitation. I'm pretty nervous because most of you know more about modeling than me. So I'll try to show at least what we've been doing in Institut Curie and how we can use Boolean models to treat some questions related to cancer. But I think it can be broader today and be put in a different context. I wanted to concentrate on heterogeneity, and I will show you how. So I work in Institut Curie. Actually, we work in the old office, close to the old offices of Marie Curie, and you can still visit the offices if you come to Institut Curie. It's both a hospital and a research center, which is very handy because we have first access to data and also some nice collaboration with clinicians with very concrete questions. So I work in the computational systems biology of cancer group. And what we try to do is to study how both intra and extracellular perturbation of the healthy genome lead to tumorigenesis. And we do that using formal methods. The postulate that we start with is that to link genotype to phenotypes, we have to go through networks. It's one way to consider cancer. It's a network disease, and in fact, what it means is that you could imagine that wild type is a certain network, and when you have some perturbation, it's some kind of rewiring of the network. And this is really what we start with. This is the initial postulate where we try to translate the knowledge into networks. And of course, we put on top of that dynamic layers to be able to reason on that. So this is the first postulate we start with. There are many challenges when you want to model cancer, of course, and again, one of them is heterogeneity. And it comes in several forms. You have different types of heterogeneity. You can have interpatient variation, intra-patient variation, and intra-tumor variation. The interpatient variation is, even though you have the same cancer, each patient will have a different cancer. And what I mean is that you won't treat a patient with stage 1 disease and stage 4 disease the same way. And in fact, if you look at the molecular profile of patients, and these are genes that are very often mutated in breast cancer, and you see that in each patient, you don't have the same mutations, and you don't find two patients that have exactly the same profile. So you do have a lot of heterogeneity inside each cancer. You have also an intra-patient variation here. When you look at the primary tumor and the secondary tumor, they won't have the same profile, even though they come from the same tumor. And another one that is, I think, very important is the intra-tumor variation. Inside the tumor itself, you have several mutations, you have several clones. And usually when we look for a treatment, we want to target the biggest clone. But in fact, you have to remember that this heterogeneity may be the cause of a relapse, or even the resistance to a treatment, because if you target one of these clones, the biggest clone, then the second one will have maybe a chance to come back. So this is something to think about, of course. There are several ways and several studies that have, of course, studied heterogeneity in cancer. And we have a lot of hope with single-cell RNA-seq data analysis, because we can identify also the different cell types that are playing a role in different cancers. So these are some of the important aspects. But I want to, and especially for this audience, I want to also say that heterogeneity may also confer an advantage to fight against cancer. Of course, you have the T-cell heterogeneity, for instance, is essential in the context of the immune response, and we know that the immune system is playing a very important role in cancer. So this is just a side note. So again, we have a lot of genetic heterogeneity in cancer, and I'm mentioning it genetic because there are also epigenetic heterogeneity, but I will focus today on genetic one. So how does it work? You start with a cell, it divides, and across the different divisions, you may acquire some mutations, some random mutations. If the system is doing its job, if the body is working well, then these mutations, if they have an impact, will be suppressed from the system. But you know that with cancer, what actually happens is that the body escapes these safe mechanisms, and the cells that should be removed or remain. And they are passed on to the next generation, where you have more mutation and sometimes some extracellular cues also that will create some new mutations. And at the end, what you have is a very heterogeneous mass of cells and with different profiles. So the tumor is composed of multiple clones. This is something that we agree on. But again, they are all different, but each of these cells will have a different cell fate depending on the mutations that they have. And this has to be also taken into account, that one clone will have a certain cell fate and maybe another clones will have a different cell fate. So this is something also to consider. Of course, the tumor doesn't work by itself. It's in microenvironments where you have pro and anti-tumoral other cell types that can favor or fight against this the growth of the tumor. So this has also to be taken into account, and I'm sure that all of you agree on that. At the end, and this is a consequence, patients with the same cancer will respond differently to the same treatment. So we take all these aspects of heterogeneity and construct that use that to construct, a mathematical model. As I mentioned earlier, this is our initial step. We start by formalizing the knowledge into a network, then translating this network into mathematical terms that we try to parameterize with the data that we have and simulate what is already known about the system, and only then we can suggest some predictions. But again, this is pretty standard. As I said, there are several aspects, and I would want to start by focusing on modeling one cell. And this is a choice. I mentioned this is very heterogeneous, and you have to consider the microenvironment and other cell types. But before, you have to know what happens inside the cell. So this is the first step, looking at one cell. And what we are going to do is take this cell and describe the signaling pathways inside this cell. We want to do that because we want to be able to evaluate the impact of a set of mutations on the different cell fates, different phenotypes that can be observed. And we want to be able, in these conditions, to suggest some intervention points, what we need to do, where we need to intervene to revert the phenotypes, the diseased phenotype. And again, all the intercellular interactions are ignored, and we fix the inputs to define some extracellular context. So presence or not of growth factors, presence of not of death signals, and so on. And the mathematical formalism that we do to do that, to describe this individual cell, this single cell, is a logical approach or a discrete approach. This is the type of networks that we deal with. We have a regulatory network or influence network, which is one of the 3 SBGN type of networks that are defined. And what it is a set of nodes that can be proteins, genes, complexes, anything that is important to describe the system. And you have directed and signed arrows between these nodes. And you have positive when it's a positive influence, and a negative when it's a negative influence. So it can be degradation or anything that any meaning it can have behind. Each of these nodes can have 2 values, 0 or 1. Actually, it's a variable at this point, not a node. It becomes a variable, and they can take 2 values, 0 and 1. And we will define the conditions when it goes from 0 to 1 with these logical rules. And so what it means here, this logical rule here for C, the condition to go to 1 will be either you have a presence of A or the presence of D, and always the absence of B for C to be active. And we do that for all the nodes of the network. And then we look at the solution space. And we usually build that as a state transition graph, where each node of this different graph is a vector of all the stages of all the nodes of the system. And the arrows will be the next possible states, depending on these logical rules. And at the end, what you have when you can no longer go anywhere, you have stable states, stable solutions. You can imagine that constructing this graph is very difficult when you have a lot of nodes, because it's 2 to the power n, and it's it can become very difficult to describe it, or to describe any big network. So what we did about 10 years ago with Gauthier Stoll, we developed an approach which is based on Markov processes. And what we did is, rather than constructing it, we decided to build it slowly and travel through it. And doing so, we are able to give a transition rate, to give some timing in the activation. So to go from 0 to 1, you can give a time. So you can associate some kind of continuous time to this discrete framework. And same thing for the K down. So you can also give a time for going from 1 to 0. That way you can also give some initial conditions, the probability for one trajectory to start with the value 0 or the value 1. And then to construct this state transition graph, you need to have enough trajectories to be sure that you cover the whole state space, because you will start with one trajectory that will go like this, and then another one that will go like this, but you want to make sure that you cover all of it. And this K up and K down, in fact, will give you a higher probability or a lower probability to go from one node or to one node of this state transition graph to another one. And this is very handy, because you can associate to each of these nodes a probability and follow it over time. It's something in between a complete discrete and coarse grain model and a continuous model where you would have to give all the parameter values. And it's one way to quantify the perturbation, because if I do a mutation in this network, the state transition graph will be different, and the values for the solutions will be different. The probabilities to reach this stable state will be different. So I hope I was clear, but we can discuss about this afterwards. I would like to give you 2 applications of how you can model that, and I'll try to be quick. I see that time is running. The first one is a bladder cancer model that we built a while ago now, but I wanted to show you what we can do also with only mutation data. So the question that we asked at the time was about the effect of mutual exclusivity or co-occurrence between genetic alterations. And in bladder cancer, you have actually 2 pathways, 2 different tumor types, actually more, but you have big families of tumors that are not driven by the same mutations. And we wanted to see if with some data and some statistical analysis, we could understand and validate these 2 tumor progression pathways. So it's not signaling pathways, it's progression pathways. So we started with in-house data that we had, about 178 patient data, and we both had CGH and mutation data, and we decided to focus only on these genes, the genes that are related to cell cycle entry and some MAPK nodes, some MAPK genes. And we did a simple statistical analysis, a simple Fisher test, where we tested if they were 2 by 2, if they could show some mutual exclusivity or not. And we came up with 9 associations that we found in our data and that we checked in other data sets. And we could have stopped that. We could have stopped there, sorry. But we decided that we wanted to explain why we would see this mutual exclusivity or this co-occurrence, and if we could give some kind of mechanistic understanding of these associations. And for that, we built this network and this model that extended a little on this list of genes. In the wild type situation, it's pretty deterministic in terms of apoptosis, because you have, if you activate DNA damage, you actually go into apoptosis. And then you have also possibilities to go to proliferation and growth arrest. And you have these probabilities in the wild type. And then we simulated all the associations. And for what concerns, I give 2 examples here. For what concerns the mutual exclusivity, we looked at FGFR3 overexpression, which is a growth factor, and E2F3, which is a transcription factor for cell cycle genes. And we had a proliferation here about 60% and 66% for E2F3. When we combined these mutations here, the probability for the double mutant didn't change much. So we concluded that there's no real advantage for the tumor. So it's P is for proliferation. Maybe because they are in the same signaling pathways. So if you have, maybe one is upstream of the other, so there's no real choice or advantage for the tumor to do 2 mutations that in fact don't matter. Or maybe they are in different tumor progressions. For the co-occurrence is a little different. So we did also FGFR3 and PI3K. And in this case, we see, when we do the combined mutation, we see a slight increase in proliferation. Maybe they are in parallel signaling pathways, and this is why we see this advantage. But it's still not much advantage. So we chose to look for a 3rd mutation that maybe would increase the proliferative potential of this tumor. And we did an automatic search, and we find CDKN2A knockout, and in this case we have 100% proliferation. So we concluded, we looked into the data then to see if these 3 alterations indeed had a meaning, and the patients in our data set that had the 3 alterations were all invasive. So we concluded that maybe FGFR3 and PI3K mutations prepare a good context for invasiveness, and the 3rd one will be very dramatic for the patient. So this was a small model that we built, but we showed that even with simple statistical results, we could go a little further and suggest some mechanistic understanding. But very recently, we decided also to include some continuous data, so RNA-seq data, and personalize the model rather than look at some generic combination of alterations. And this is a work we did with Ana Montagud in Barcelona and Julio César Rodriguez in Germany. And this is a paper that was out not so long ago. The question was to suggest personalized treatment to prostate cancer patients. And same thing, we constructed a Boolean model, a little bigger this time, because we wanted to include a lot of pathways that are altered in cancer. And we simulated some normal conditions. If you activate growth factors, you're supposed to see proliferation. And then we took some omics data. We took a TCGA data where we could have discrete data and continuous data. And also we had information about the severity of the tumor per patient. And then we developed a method called Profile, where we actually try to integrate this omics data into our model. For mutations, so for discrete data, mutation copy number alteration, if you have an activating mutation, you force the value of the node to 1. If it's an inhibiting mutation, you force the value to 0. Very simple. For the RNA-seq and proteomics data, it's a little different. We change the transition rates rather than change the value of the node itself. So the rationale behind is that if you have a higher expression of a gene in a patient compared to another one, it probably means that it activates faster. I mean, this is one rationale, one idea. Difficult to validate, but this is something we tried. And then what we have at the end is one model per patient and with their own parameters. So each of these patients have their own parameters. On each of these models, what we did then is we searched for optimal treatments. So for each of these models, we systematically inhibited all the nodes of the models, and we selected the nodes that significantly decrease proliferation, so like the treatment would allow the decrease of proliferation or increase apoptosis. And then you can make some conclusions on the whole population, but you can also make some predictions on individual patients and suggest some possible combination of inhibitors for this patient in particular. We know in the clinics this is not very possible to suggest personalized treatment, so you can also look at group of patients and suggest treatments. And then what we did next, and I won't show the result here, but we did some experimental validation of some of the candidate targets we identified in a sensitive cell line LNCAP. So this is it for the individual model. And as I said in my introduction, it's not alone. You have to consider the microenvironment as well. And ideally, what you would do is you would construct for each of the cell types of your environment, you would construct a model. And we know that there exists. We know that there are models of tumors that have been developed, some of T cell and some of the work of Thomas also. Thomas Helikar could also fit some of these models. You have models of macrophage as well. So with this in mind, we collaborated with Paul MacLean in building PhysiBoss, which is an agent-based modeling tool, a PhysiCell, that takes into account intracellular description. So the individual models I explained, you could actually feed them into this agent-based model, where each of these agents would be a Boolean model. As you know, agents in this context can diffuse, move, proliferate, die, interact with the environment. A cell has a size, a position, and some dynamical properties. And let me show you an example, and this is taken from the paper of PhysiBoss that we published in 3 years ago now. It's an example of TNF treatment. This is a very simple model where you have, it doesn't really matter what's inside, but what you can understand, what you can look at is when you activate some death receptors like TNF, activated by TNF, sorry, you can either go to survival or to death, either by necroptosis or apoptosis. And this is the model we started with. What is important to see is that, so you have this individual model, but you also have a possibility to interact with other cells where NF-kappa-B can amplify the TNF signal and promote the activity of TNF in the neighboring cell. So keeping that in mind, we put that into the PhysiBoss model, and we then tested, we did some treatment. First, the control, when you don't have any TNF, the tumor grows. It's in condition when you have enough nutrients and enough oxygen, anything you want to grow, and the tumor grows. When you actually put a TNF treatment, you see that the cell doesn't grow as much, and a lot of cells can die by apoptosis or necroptosis. Another thing you can do on this with this tool is to test clones. So here, this is an example of a clone. You have 75% of cells that are wild type, and you have 25% of cells that have these two mutations. In this context, if you have this clone, then you can put TNF, but actually the tumor will continue to grow, and it won't be as efficient as the wild type situation, as I showed earlier. I won't go too much into details, but what I want to show is that you can do some in silico experiments, and you can see if the tumor grows or not in certain conditions. You can test the effect of a treatment on some populations that include clonality. And I didn't show it, but you can also try some pulses or continuous treatment and see the effect. And in fact, if you do continuous treatment, the cells will show resistance. With pulses, it would not. So I showed here some example of MABOS and PhysiBoss and Profile, but we also have some other tools that we can talk about. And my my presentation is done here, but if you allow me 2 more minutes, I would like to give you 3 slides of a possible discussion with the working group tonight. The first one is a work we've been doing on immunogenic cell death, and our tool is UpMaBus. It's a little like PhysiBus, but without the spatial aspects. It's more the idea of ODEs, where you have like a mixture of cells and you don't really know where they are, but they can interact. And we applied that to immunogenic cell death, where you actually give a chemotherapy treatment, and this induces some cells, some tumor cells that will die, and then will activate the immune system. That will lead to, by activating dendritic cells and so on, that will lead to the activation of CTL, and that will kill the remaining tumor cells. Some chemotherapeutic treatment are not as efficient as others, and we want to understand why. And for that, we build a small models where we have cell types here as nodes, and then interactions between these cell types and the differentiation also of CD4 into different T cells. We simulated populations, it's not gene expression now, but it's population of cells, and we could do some predictions, but this is something we can talk about. The second thing is something that Vincent and Marco will be able to talk about a little more, and they are in the audience today. It's a PhysiBus, as I mentioned, and the application to COVID, and Vincent has been very much involved in COVID development model. This is a very early model, but he has done a lot of work on this since then. And then the last one is the profile. I mentioned it, but I want to mention it again. It could be one tool towards the digital twins that you guys work on. We can use this analysis and improve it, of course, to include different types of data. We can use this data to define the cell types and their size, initial population size, and use this idea also to personalize better each of the cell types, which as of today, it's kind of difficult to do. And that's about it. So these are just ideas of where we could go in the discussion. And these are the tools and the people that I've been working on. And I can stop now.
Well, thank you very much. That was a lightning overview of a large amount of research. So I appreciate you going over that. Okay. The floor is open for discussion.
Should I unshare my screen maybe, and I can share again if.
Okay. Is it Navid? Please go ahead.
Yes. It was a wonderful talk. I have a question. I'm curious about when you transition from the Boolean model to the more spatial kind of setup using PhysiBus. I was wondering how actual spaces contributing to this model. Is it like based on like cell-cell contact, or like is it like, I don't know, like a weight that like weighs the inputs that you put in your Boolean model? I just wanted to know how space contributes in this setup.
So I'm not sure, sorry, I couldn't hear very well the beginning of your question, but the way it works, PhysiCell is already an agent-based model where you can actually simulate already cell-cell contact and the effect of the environment or whatever is in the environment, and if you're touching the extracellular matrix or not. And PhysiBus, MaBus, the MaBus model, what it does is it's linked to some parameters that are already included in PhysiCell. And then depending on some values that you have from PhysiCell, it feeds the model, the MaBus model. You see, you have a trajectory, and the output of MaBus would then feed back on PhysiCell. I'm not sure I'm answering your question, but I didn't hear. It
answered part of my question, but my main question is like you talked about the heterogeneity of T cells and cancer cells and all the other immune cells. Well, in a microenvironment, they're like distributed everywhere, right? They're not like all like uniformly distributed. So like, for example, one cancer cells can be very far away from one cytotoxic cell or very close to a dendritic cell or things like that. So I want to know how this distance and like this location of cells contributes to this model.
So actually, maybe Vincent and Marco can answer better, because the idea is that you have to distribute them initially. And I'm not sure I know how it is distributed initially, because what we've been working on so far is really the growth of a tumor. And not so much including different cell types. So Vincent has been working with that on the COVID model, so he can answer better than me.
Okay.
Yeah. Then, so yeah, to explain how cells can interact at a distance, for example, then you will have either cells traveling toward each other. So one cell will, for example, be attracted by the other cell by some gradient of protein that she's secreting, or they can just communicate at a distance using the same kind of signaling molecules that cells will be secreting and other will be sensing.
So there's like a background gradient of like molecules that is like the underlying communication system in this model.
Exactly,
right?
exactly.
Okay.
You can, in this framework, you can just create multiple substrates which will diffuse and interact with each other and with cells.
Awesome. Thank you. Thank you so much.
And what we want to do also next is also personalize these models, these individual models, and so you add a layer of complexity here where you have to personalize the model inside the cell and then distribute them everywhere. It's complicated.
Yeah, I can
but
imagine.
we're going towards that, and we're trying to find ways to also take into, and I didn't talk about that much, but we also take into account families of models, and not just one single model. That, I mean, the logical rules, there may exist many models that actually fit what you want to describe. So we also take that into account and try to include that in PhysiBoss. So again, another layer of complexity.
Thank you. Thank you so much.
Okay, other questions? Tom.
Yeah, so related to this conversation, so the linking between the Boolean model and the agent-based model, how do you? So I'm assuming you have some predetermined threshold. Say, you know, apoptosis has to be 20% before the actual agent dies. Is that right? And how do you determine those thresholds or parameters on that side?
So there are some parameters that are already set in PhysiCell that we use, and then I think Marco has been working a lot on the model of invasion, and he's trying. It's trial and error. We try to do some sensitivity analysis on some of the parameters and on the threshold, but we have to do them one at a time for now. Because there are many parameters. So we try to use Boolean models to simplify the system, and then we put back into a more complex system where we have parameters to fit. But maybe he can tell you about it, but we're also exploring the possibility to work on surrogate models to deal with this, defining the right parameters, the right threshold, the right parameters that we have in the model.
Yes, if I can jump in for this question, hope you can hear me.
Yes.
Just to come back to what you asked, Thomas, at the beginning. Yes, basically the output of the Boolean network are linked to some behavior that are coded inside the PhysiCell PhysiBoss. And so when you have apoptosis as phenotype, like in the example that Laurence showed, then you trigger apoptosis also immediately in a simulation. Otherwise, another thing that can happen is that one phenotype is linked to a variable inside the multi-agent based framework, and when the variable then reach a certain threshold, or maybe the variable is linked to another behavior, then it triggers something else. And for what concerning the threshold, as Laurence said, we were thinking about the possibility to use surrogate models to train a deep learning algorithm in order to, let's say, resimulate what Physibus is doing, but in a faster way, and then run lots of simulation with the surrogate models on a small subset of the parameters. So in this case, some thresholds of interest that we want to fit, and then every time that we have some interesting results from the surrogate models, we will reproduce it with Physibus. And then if the result is acceptable, then we keep it. If not, then we will retrain the surrogate model based on this comparison. So there is this double link. And of course, when the surrogate models prints the output, when you have the output of the surrogate models, you compare it with actual data to see if what you are simulating with the surrogate is actually valid. So this is our way to we thought of this way to fit the parameters.
Yeah, it's not done yet. It's something we
No,
plan
yes.
to do to fasten the process. But...
It's never done. There's always more to do. And so you need surrogate models because you are mapping now the entire state space to calculate the transitions.
No, we need it basically because the Physibus simulation are getting too long. And so evaluating every set of parameters just takes too long. So we are hoping, basically, we are betting that retraining regularly the surrogates and evaluating them will took less time than just evaluating the Physibus simulations.
But again, I'm sure we'll run into some issues with these that we don't think about yet. But this is the plan. Yeah.
I was going to ask a quick follow-up on that, which is if you have a threshold, you're effectively saying that the probability of transition is 0 or 1. But maybe it would be more natural in the context of the Boolean network models if the transition probabilities were a function of the values coming out of the MapBoss model. And so instead of saying that you have the cell die when the value of some signal reaches X, you say that there's a Hill function of that signal which controls the probability of death per unit time. And then you basically would be bringing that Boolean transition probability into the spatial model as well. I wonder if that's a problem, or if there's a reason or no advantage or disadvantage to doing it that way.
So the probabilities of the Boolean models should not be taken with so much faith because they depend on the structure of the network. So if I have the ending probabilities will depend on the structure. So if I developed a model with a lot of nodes in the apoptosis, I will have more probabilities to reach the apoptotic node than the survival one. So I try to be very careful with the probabilities you get from the Boolean model. It's more the comparison that is interesting and important than the value itself. But yes, I mean, of course, we can once we're more comfortable with the structure and the equilibrium of the nodes inside the network. Of course, this is something that could facilitate the transitions and the exchange with the extracellular environment.
Okay, other questions? Navid, do you have another question?
Yeah, I have another question. I think it's mostly experimental. At some point, you mentioned about the cytotoxicity assay. I was curious about whether your model tried to confirm the outcomes of the assay, or you use that to calibrate your model. What was the use of that?
No, it was used to confirm the predictions. But again, I don't want to be quantitative. I mean, it was really either it's working or it's not working. the drug is effective or it's not effective. I'm not, yeah, but it was to validate. Yeah.
Right.
So we did all these experiments on the cell lines with the personalization of on the cell line, and then we did the automatic inhibition and so on, and we selected only 2 nodes that we tested experimentally.
Okay, thank you.
Yeah, is cancer one of your interests? Is it? You said you have broad interest in the working group. Is it also something you explore?
Well, certainly the modeling approaches you're using are very relevant. Mm-hmm. And as you see, a lot of the signaling pathways that you're looking at are ones that are important in immune response as well.
Yeah.
You have NF-kappa B, you have apoptosis, you have other kinds of cell death, you have immune hiding of the cancer. So there are many pathways that are shared.
So we're not expert in the immune system yet, but we are exploring more and more, of course, the impact of the immune system on treating patients. Of course, we have also a model. I didn't show it, but we have a model of the effect of anti-PD-1 and anti-CTLA-4. But it's really very high level. I mean, it's not very detailed as you guys would be interested in, I think.
So if I were to add to the description that James just gave, I think that the way the working group has evolved, the interest lies generally in multi-scale modeling as applied to personalized models. And there could be models of infectious diseases, the immune system, but sort of generally, certainly my interest, and I think that of a number of working group members, has evolved to our construction of digital twins of various sort, to use a buzzword. And so I think what you've been talking about fits in extremely well.
Yeah, I mean, we're also I put a digital twin just for you in the discussion slide, because this is something we're interested in, of course, as well. But I have to say that the methods we have for personalizing the models, I have no way to validate them. And for me, this is the biggest issue. So I'm presenting the methodology, but I cannot tell you that this is good or not. I know it's coherent with some of the results when we do some survival data, survival analysis comparison. We see that it goes in the right direction, but I have no way. So I would not dare to say that we are going towards digital twins, but I would like to.
Well, I think it's an interesting topic that most people don't know, how to do this. Validation is a huge issue, and it's not so dramatically different from, you have a new drug candidate. How do you validate? How do you start giving it to people, right? And it's the same sort of issue.
Yeah, and we're not very quantitative either, so. We're not so digital. It's not a digital twin because we like these quantitative aspects.
Well, you got zeros and ones.
Yes, we have probabilities of zeros and ones. and actually with Physibus, what I like is that we can also match population size. So we can start at least with a proper population size, and we've seen that in the immunogenic cell death model, that if you play with the population size, you won't get the same results. So if you increase the macrophage, then you will get to death faster. Of course, I mean, it's obvious, but it's something we can play with on more complex models. Dendritic cells, no, I mean macrophage also, but in this model we have dendritic cells rather than macrophage.
So can I come back maybe to that performance of Physibus? Because as we talk about digital twins of whatever of immune system, for example, that's many more cells, many times more each cell type, right? And performance of these technologies is going to be key. What's the bottleneck with PhysiBOS or PhysiCell? Are you calculating steady states or attractors each sort of time step or a time unit of the agent, or are you doing simulations of the Boolean model inside? What makes it so much more computationally intensive?
Vincent, maybe you, I mean, we simulate the model, but yeah, Vincent, go ahead.
Yeah, the biggest part for PhysiBOS is actually not the MABOS part. The MABOS part is really simple. We just compute one single trajectory per cell. So it's really just evaluating one Markov chain for each cell. But still, like Marco can confirm for me, but the biggest greedy part of PhysiCell is the substrate diffusion. That's the part where most of the work is done.
Okay, so it has nothing to do with the nesting in Boolean models. It's inherent to PhysiCell.
PhysiCell.
Yes.
I see.
In fact, if you, sorry.
No, go Marco.
Okay. No, actually, if you increase the number of substrate that you want to simulate, and also if you increase the number of cells, that's the heaviest part. I mean, simulating the uptakes of molecules and this kind of things. So that's it's computationally expensive, not MABOS.
Yeah, I wanted to say that MABOS actually is a very light part to simulate signaling like this. It's something very simple actually, and it scales a lot better than say ODs if you want also to model signaling.
No, and that way we can have a lot of nodes inside each agent also if we want.
Okay. What is the ceiling of on the number of cells that PhysiCell is able to simulate in a reasonable time?
I think people from BSc went up to 10 millions, but it was using a version of PhysiCell which they developed, they adapted, which is parallel and can use several machines using MPI.
Okay.
So we are still, and they are still working on this version of PhysiCell. So we are trying to see what the limit is, but probably in the 100 or 1 billion cells, something like this, for very, very large clusters.
Yeah, there's a comment in the chat about CompuCell 3D. Also, MABOS is also included in CompuCell 3D, and apparently it's also not very costly to simulate the MABOS model.
We all have the same issues.
What other questions does anybody have? James had to leave for another commitment, so.
I think at least you know what we are doing, and especially I think maybe for some applications related to the COVID example. But I would like to have, as I said, a model of the tumor microenvironment as well. In this sense, I think we could talk. We can mature this, and then we could talk about some ways where we can converge also on this type of modeling. So including the immune system in our modeling framework would be great. And we are not very good in this, so we are very much interested in what you guys know and can suggest also.
So Laurence, you might know that we're organizing a workshop at the Institut Pascal next May. The local organizer is Ananiakis,
Ah, yes, yes.
Yeah, and it's going to be on digital twins. There's lots of room, and we hope that people from Curie will join us. Emanuela knows about it. So.
Yeah, okay, I didn't know it, but yes, I would be delighted to join. I will tell Anna to tell me.
Yeah, it's a 3 week.
Tell me the details.
Yeah, the last 3 weeks of May.
Okay.
You'll get tired of us in 3 weeks.
Sorry?
You'll get tired of us in those 3 weeks.
No, never. Never. Okay, good. Yeah. This is so. It's an opportunity to meet. I won't be in ISMB, but if we can meet in Paris, it would be great.
Sounds
good.
Great.
Yes. Yeah.
Well, thank you very much, Laurence. This was great.
Thank you. Thank you for the invitation, the discussion, and yeah, we are happy to continue the discussion afterwards.